So good morning everyone, and apologies for the slight delay in getting started, but we are now live and welcome to our webinar today. My name is Liz Harley. I'm the Content Manager for Front Line Genomics, and I'm really excited to introduce today's presentation, which is called Think Big, Screen Once, Genome Scale Editing with Custom CRISPR Libraries, and we're doing this in partnership with Twist Bioscience. Our speakers today are Charles Joseph from Twist and Edward Perello from Desktop Genetics. And rather than do too much of a lengthy introduction, I'd like to hand straight over to Charles for our first presentation. Over to you, Charles.
Great. Thank you very much for this opportunity, and thank you all for attending. So Twist Bioscience is a synthetic DNA service company, and we have a transformative platform that allows us to write DNA on a silicon surface. And in the CRISPR world, this DNA can be used as guide RNA templates for gene editing or as donor DNA for gene knock-in experiments. But we don't, when you're working in the CRISPR screening workflow, you go through the design process where you're targeting, depending on your whole, whether it's a whole genome screen or more targeted approach, tens of thousands of genes or hundreds of genes. Then you would work to build your CRISPR library and then screen and select for the desired function. But you don't just get there in one step. You have to go through this iterative process, and challenges can occur in developing effective guides that maintain specificity from your design to package clone, and so that you can maximize your screening efficiency, and you don't want to lose guides along the way and potential hits that may have dropped out. Getting over to synthesis and how we could, at Twist, create a tool that you maximize your screen. We start off with a functionalized surface with a free hydroxyl group and couple this with a phosphoramidite to create a highly reactive phosphite bond. Unreacted sites are capped to prevent non-specific reactions while oxidation takes place and a phosphate group is created. We then deblock and allow the chain to grow as we repeat the cycle until a full chain is synthesized. Traditionally, this is done in a 96-well plate, and it requires many cycles and a long turnaround to generate a large number of oligos. Another popular method for making oligos is with the use of microarrays. Oligos are made on a glass substrate using the same phosphoramidite chemistry with a new base added each round. This method provides an inexpensive way of generating pooled oligos. However, if you want to create smaller pools, you are limited by the microarray size or required to amplify out subpools of oligos. And this process can introduce PCR bias, which will then be affecting your screen downstream. But at Twist Bioscience, we've developed a novel silicon platform with the same footprint of a 96-well plate that could generate over 1.1 million oligos in a single run. This transformative platform allows for the generation of a highly uniform and highly specific oligo pools that can be used to generate a CRISPR library. And that's where we channel the power of our technology towards the application of CRISPR and can provide oligo pools. So through a partnership with Desktop Genetics, we're able to offer library designs made easy. Edward Perello will be talking more about their library design philosophy and how they combine several algorithms to really deliver an optimal library. But the pools that we're able to generate allows for precision gene editing of target loci. We have a highly accurate synthesis with a 1 in 500 error rate, and 100% of oligos in the pool are represented. And we also enable the maximization of screening efficiency with over 90% of oligos represented within 4x of the mean. We did an analysis of our pools from synthesis, and we took a look at a pool of 29,000 unique oligos from 240 clusters of our chip. And for this specific pool, 99% were within 2x of the mean, and 100% of oligos were present. And when we looked at the local uniformity at a cluster level of each well of our synthesis platform, you could see that the same uniformity is present both globally and locally. Similarly, with that same pool, we did analysis of accuracy and found that our error rate was on average 1 in 500 base pairs. And this is with off-the-shelf chemistry, the standard phosphoramidite chemistry that I explained a moment ago. Without further optimization, we're continuing to work towards improving this. And again, not only on a global level across our whole silicon platform, but at a local level on a cluster basis, we're able to determine that error rate was consistent. Synthesis GC content is always a concern for generating uniform synthesis, but we've found that across a broad range of GC content of oligos, from 15% to 85%, there was a minimal impact of GC content. And so in a CRISPR workflow, you could take an unamplified oligo pool that we've generated and then work with it downstream to PCR amplified and clone it into your vector system of choice. And in the future, we're also working towards developing a cloning-ready solution. One aspect of working with an oligo pool that is sometimes overlooked is PCR. It's a very simple technique that everybody has a lot of experience with, but there are some considerations that need to be accounted for when amplifying a highly complex oligo pool prior to cloning, because you want to follow a protocol that minimizes the introduction of PCR errors, maintain a very high oligo representation, and minimize the PCR-generated bias. So to take a look at the PCR effects on oligo pool uniformity and representation, we took an investigation of a 10K oligo pool with oligos that were 100 nucleotides in length. They contained a 53 nucleotide randomized region with GC content varying from 20 to 80%. And we took a look at PCR amplification using two different market-leading high-fidelity enzymes and did a QC at 6, 8, 10, 15, and 20 cycles, and then sequenced the pool to see how the representation changed between the different enzymes and as well as at the different cycle points. So as you can see here, looking at polymerase 1 versus 2, while they're both high-fidelity enzymes that are marketed as having a good tolerance for GC content and different oligo lengths, we see that over 6, 10 to 15 cycles, the peak starts to get broader for polymerase 1, and you're starting to get a lot of oligos that are represented much lower and the uniformity isn't as tight. Whereas in polymerase 2, it's able to maintain a relatively tighter formation across that cycle range. But again, I would just like to note that this is polymerase 1 and 2 for this specific pool. The polymerase 2 might not necessarily work the best all the time based on your oligo design and the complexity of your pool. Looking at that data in a little more detail, we see that for both polymerases, as the cycle number increases, the percent of oligos that are within 1.5 times and 2 times the mean concentration starts to drop off as the cycle number increases. For polymerase 1, 92% is within 2x of the mean at 6 cycles, but you lose 10% of your oligos as you move out to 15. And similarly, there is a drop-off for polymerase 2, while in this specific case, it was a little less. But it is something to consider when you're working with oligo pools. You want to keep the cycle number lower if you can, and just start with the minimal amount of material that would take you into your cloning workflow so that you don't generate any bias and lose sequences. Looking at the impact of overamplification on sequence dropouts, we see that as the cycles move from 6 to 20 cycles, in the 6 to 8 range, there's a very small difference in the fraction of the population that is 10% below the mean concentration. Whereas if you move from 10 to 15 to 20 cycles, you see that there becomes a more significant dropout in sequences. If we're looking at the effect of overamplification on GC content, the black line represents what would be the same representation for 6 and 20 cycles, and any of the dots and oligos that are above the line are overrepresented at 20 cycles compared to 6, and all the dots below the line represent oligos that are underrepresented at 20 cycles versus 6 cycles. And as you can see, that there is a bias that starts to be generated for GC content when you're either overamplifying your sequences compared to the 6 cycle mark. So just to recap that, enzyme selection and reaction conditions really affect oligo representation and uniformity. We found that generally 6 to 10 cycles of amplification can produce sufficient material for cloning without significant PCR effects, and a higher number of cycles can introduce more GC bias and sequence dropouts, but we always recommend that our users empirically determine what the best PCR conditions are for their own assay, because again, it's very pool dependent, sequence dependent, and it'll also depend on what your cloning strategy is and the organism you're trying to express in downstream. So looking at a real-world example, we've worked with Kosuke Yusa from the Sanger Institute, who was working on creating a whole genome library, and in his pool, he had approximately 5 guides per gene in the human genome to get 100K complexity for their CRISPR library. And what they've done is that after receiving a pool from Twist, they PCR amplified it, cloned it into a lentiviral vector, as well as prepped it for NGS sequencing, and they did an analysis of an NGS analysis of guide representation of their pooled sequencing. So they did a comparison of an array-based competitor oligo pool with a Twist pool, and we see that for more than 99% of their guides, there's a wide distribution. We see a range of 6 logs, and there's also a very long tail of low coverage reads. And a lot of these, when you're trying to express them downstream, could get dropped out of your screen, and you risk losing data that you would have gathered from those guides. When we compare that with the Twist oligo pool, the higher uniformity of synthesis enabled a tighter distribution of reads across the entire library with a 4 log difference, and it did not contain a long tail of low reads as well, so that more reads per guide were represented so that you could get the most efficient screen from your workflow. So through the synthesis that Twist is able to generate, very high quality uniform pools with very high sequence accuracy, so this will allow you to maintain your guide specificity and maximize your screening efficiency. And to cover the challenge of library design, we have partnered with Desktop Genetics, who is a bioinformatics company specializing in CRISPR design. And with that, I'll hand it over to Edward Perello, who will talk to you a little bit more about what their CRISPR library design service could offer, and as well as their approach to designing libraries.
Wonderful. Hello, everyone. Can you hear me? I'm going to assume that everyone can hear me. Wonderful. So thanks very much for that excellent introduction, and thank you to everyone at Twist. It's always good to be working with you guys, as well as everyone over at Front Line Genomics as well. So I'd like to start with a brief agenda on what I'm going to be discussing with you today, which will be a brief introduction to my company, Desktop Genetics, and I'm going to tell you a little bit about how we support researchers with their work using CRISPR in the lab. First, I'm going to talk about the DeskGen Cloud software platform, and then I'm going to go into a little more detail about the custom CRISPR library design services that we offer. And I'm then going to wrap that up by exploring the CRISPR design dogma through a study that we conducted with one of our partners last year. So moving on to slide 3, just a little bit about the company. So as Charles said, we are a CRISPR design specialist bioinformatics company, but we have two flagship products and services. The first is the DeskGen Cloud platform, which is essentially an amalgamation of many of the well-known, well-respected sgRNA design tools that have been published in the academic literature, brought together in a single place, optimized and wrapped into a wonderful graphical user interface, really to present people with everything they need to get started with CRISPR for very small, highly targeted experiments. The second service that we offer are our genomic services. And here you're really leveraging our expertise of working in CRISPR for the last 4 years, where we can design a custom CRISPR library. We can also work with partners to conduct that experiment on your behalf. We can also analyze the data that is generated from that experiment. And genomic services really falls into two separate sections, the custom CRISPR library services, but then also CRISPR genotyping and analysis, which is more to do with that analysis. So on to the next slide, DeskGen Cloud. So many of you may have come across this before. If not, I'd certainly encourage you to sign up for a free account at deskgen.com. And really, we've been inspired by many of the academic scoring functions and software tools out there. And we know that there's just a tremendous amount, and it can be very, very hard to keep up with the latest CRISPR research. And so we try and put that all in one place for researchers. We can give you access to multiple nucleases, whether it's CPF1, SPCAS9, or the nickase versions of SPCAS9. And we can allow you to change protospacer lengths. There's a dedicated mode for knock-in and knock-out. We have up to 10 scoring functions, which can be accessed through the guide picker tool, all of which I'm just going to show you very briefly. So on slide 5, you can see an outline of the knockout mode. And so here what I've searched for is the CDC20 gene, and we're showing all transcript variants with some predicted promoter and terminator sites. Here you can see in the second section, there are 2 guides that are open, which are those little yellow bars, and each black lollipop that you see in the center of the screen represents a valid guide site, and embedded within it is the DENCH 2016 on-target scoring function readout for that particular guide. And then down towards the bottom, you can filter those guides. There's a little filter which will allow you to select for guides above a certain on-target threshold. And you can then select a guide and run an off-target scoring search. And here we're running the exact same scoring function that you would see on the MIT CRISPR tool. The only difference being the readout, which if you move to slide 6, you can see that this is a little bit different from what you may be used to if you've used the MIT scoring function, where we're presenting you with an off-target analysis, which presents you with the number of off-target hits, with the number of mismatches in each hit, as well as the location of those hits, as it occurring in a coding region or a non-coding region. And this is significant as it impacts the overall off-target score or the specificity score. So the knockout tool is a really nice, easy way to design a few guides for an early-stage knockout experiment. The knock-in tool is, of course, the analog to this. It's a slightly different graphical user interface where initially you set a focal point, as you can see on slide 7. So you enter in the gene. Again, I've searched for CDC20. And here I've selected one of the transcript variants at the top in blue. And if I proceed onto slide 8, then you can see that we have another very similar layout where we're showing not only the gene and all of the sequence, but we're also showing the open reading frame and the peptide as well. And what you can do is you can click on one of those amino acids, mutate it, and then generate a donor. And the donor you can design according to various donor design rules if you want to go for a single-stranded oligo donor. Or alternatively, if you want to do something a little more fancy and use, say, the IGI asymmetric donor design rules, these are supported as well. Moving on to the next slide is just a layout of the guide picker tool, which we're really, really proud of. This is the latest tool that we've released on the DeskGen platform, which allows you to conduct a side-by-side comparison of various sgRNA design parameters. So here I've opened bracket 2, and I'm looking at GC content versus the percent peptide on the left. And I've then, on the graph on the right, I'm looking at the off-target score plotted against the DENCH 2016 positionless score. And we support 10 different scoring functions on this tool, and we'll be adding more in the future as we continue to evaluate them. Once again, just jumping forward, you can see how you can change the layout of these scores. Here in slide 10, I'm showing microhomology versus percent peptide. And now I'm plotting one instance of the on-target scoring function, the DENCH 2016 full, against the DENCH 2016 positionless. And there's just a really useful way of quickly narrowing down on a set of guides that you want to take forward. If you highlight guides in the graph on the left, then these will show up in the graphs on the right. And you can also adjust some threshold parameters so that you can only select guides over or within a certain score. So that is DeskGen Cloud. I now want to move on to the next slide, where I'm going to introduce custom CRISPR libraries. So Cloud is really about leveraging your own expertise in CRISPR, or giving you as much as we can initially so that you can get started. The difference between the custom library design is and Cloud is that you're effectively leveraging our expertise to design this library for you, for any size, any complexity, any experimental intent. It doesn't just have to be a knockout library. It could be a CRISPR-A, a CRISPR-I library. It could be an epigenetic CRISPR library. But the simple fact of the matter is that we'll work with you to design the right guides that target any gene or any intergenic region, and we can do this in any organism and any specific cell line or cell model that you're using. And in addition to the design that we offer, where we're really wrapping in a lot more advanced scoring functions that are not included in the DeskGen Cloud version, we can also get this library manufactured for you by one of our partners, which is, of course, why we are working with Twist Bioscience. In addition, we can offer you long-term dedicated in-house bioinformatics support so that we can help you analyze your data. And really, we just want to assist you in making sure that your design of that experiment is right from the beginning, but then guiding you through the process of the entire experiment towards the data, the analysis, and eventually the publication as well. So just in terms of a few of the options on what this looks like on the next slide, of course, this is all tied together by the computational guide design, but then we have some additional options beyond the simple knockout library or a knockout panel, which many of these off-the-shelf CRISPR libraries are geared towards. We can design a CRISPR-A library, CRISPR-I. We can target functional domains. We work with Twist for oligosynthesis, another company for a full cloning service, and another company for lentiviral packaging as well. So you really have the full optionality of CRISPR genome editing at your disposal through the company. One thing that I will touch upon in a little bit more detail later is the idea of sequencing the model that you're working with. And so we do offer a range of sequencing options for our clients, where we can sequence the specific cell line that they're working with, generate a new genome assembly, and then design a model-specific library around that. And I'll explain a little bit about why we offer this service towards the end. Moving on to the next slide, CRISPR genotyping. This is really where we go beyond model characterization. Instead of just assembling a new genome, which is the basis of a new library, we can also support you with analyzing and quantifying your gene editing events, whether that's on-target and off-target detection using amplicon sequencing, or combined detection using some of the newer and more experimental assays for multiplexed amplicon sequencing. And finally, of course, we can work with you to analyze that data. But we're all here to discuss one thing on the next slide, which is the high-performance guide design, which pairs very, very nicely with the high-quality oligosynthesis that Charles has just laid out for you. And so everything that I'm going to discuss over the next few slides applies a great deal towards the custom pooled sgRNA library service offering that we have with Twist. And now I'm going to explain to you a little bit about our sgRNA design philosophy and how we've arrived at that. So before I move on to slide 16, I suppose I should just outline how we work at Desktop Genetics. So our goal really is to enhance screening efficiency, and the best way to do that is to start with a custom-made library that is designed for your organism and for your model. We're looking at a number of things that we want to optimize for. First and foremost is the activity of a guide, so how well can it bind at a specific region and elicit a cut or a gene editing event. We're also looking to optimize for the specificity of each guide that is designed. That is to say, we do not want guide binding off target and making it difficult to have a reproducible assay. We also look to optimize a number of sequence parameters, such as GC content. And finally, we also look to optimize where we design these guides, like the target itself and the target's environment. And the long-term goal of curating a best-in-class sgRNA design philosophy is to create a machine learning algorithm, or to train a machine learning algorithm that can take any given cell, any given line, any given experimental intent, and determine the minimal set of guides required to achieve the desired experimental outcome. And so moving on to the next slide, 16, the experimental overview. I'm going to outline the essentiality library screen that we conducted with Horizon last year. So we did something a little bit different here, which was quite unusual for most studies at the time. Our overall aim was to target essential and neutral genes for knockout, but also to create a valid training data set that we could feed to our machine learning pipeline in order to arrive at optimized guide design. And we did that by designing not one set of guides, but two sets of guides. The first set of guides were really where we were challenging some of that CRISPR sgRNA design dogma, where we sought to take a lot of the published literature, really drill down into the algorithms and test them to see if their assertions were correct. But the other set of guides that we designed was a set of guides that we believed would perform poorly. And the reason for this is that when you are training a machine learning algorithm, you do want to give it two references. You want to give it a positive reference and a negative reference. And that is what we sought to do here. So we modeled our study after the Shalom et al. 2014 depletion library. We designed 3,000 guides targeting 100 essential genes and 7,500 guides targeting 745 neutral genes. And this was all for the A375 melanoma human cell line. And these controls, of course, were the neutral gene data set, but also NAG non-canonical PAM targeting guides, which we had used because the NAG PAMs have a much lower activity than NGG PAMs. We stuck with SPECIFICITY throughout this study, and our readout was based on depletion of NGS reads from knockout-induced cell death. So huge thanks to Horizon, who, of course, without working with them, this would not have been possible. So in terms of how this experiment was laid out on the next slide, 17, we've got our experimental overview, where we manufactured and cloned the guides that we had designed into lentiviral expression cassettes. We sequenced these to verify our clones, and we then packaged and transduced these and selected them with pyrimycin. And at the point of transduction, we created two biological replicas, which had good correlation throughout. And then you'll see on the right of this slide that we were conducting NGS reads throughout the course of the experiment, where we were monitoring for read depletion as a measure of sgRNA efficacy. So on to slide 18, where we present the results of our activity score comparison. So the most well-known and most widely used activity scoring function at the time of the study was the Dench et al. 2014 on-target score. And essentially, this scoring function provides a predictive measure of how well a guide will bind at a target and elicit a gene editing event, or how it will cut that target. A score of 100 suggests that the guide will, according to the literature, be very, very effective, and a score of 0 implies that that guide will be much less effective, though the predictions are not 100% absolute, but again, they are a measure. What we saw is that sgRNAs with a high dense score have significantly increased read depletion, which was very much in line with what we were expecting from the literature. And as a result of this, we began to build all of our scoring functions around Dench 2014. We knew that we had validated this for library design, and that is something that we wanted to take forward at the time, as indeed we did. We also looked at another scoring function presented on the next slide, 19, called the Chari et al. Scoring function. This is very similar in scope to the one developed by the Dench group, or the Root lab, I should say. And it was developed in George Church's lab. And again, a score of 100 implies that the guide will have a very good activity. A score of 0 implies very poor activity. And again, we saw that these were generally comparable in terms of being positive predictors for on-target activity. However, moving on to the next slide, things have changed a little bit since our study last year and since the Dench 2014 scoring function was developed. In fact, there has been a new scoring function released by the same lab, the Dench 2016 scoring function, and you can see that this really blows the others out of the water. We went back and we retroactively scored the guides with the Dench 2016 scoring function, and we saw that it was a far more powerful algorithm in terms of its predictive capacity to design a guide that would have a high activity. And you see this only in the essential genes and not in the neutral gene data set. So the Dench 2016 scoring function really has sort of surpassed Dench 2014 and 15 and is now the core component of our on-target prediction algorithm. So now I want to talk about specificity on the next slide. So specificity, as I mentioned, is about this idea of where is the Cas9 and the guide binding. And is it binding only in one place? Will it bind in many places? Will there be these off-target effects, which I really want to avoid because this could cause deleterious knockout of other essential genes, which could have an impact on my phenotype, which is absolutely something I want to be avoiding in a genome-wide screen. So the Hsu et al. Scoring function, which was the first CRISPR scoring function, which many of you would have used in the MIT scoring function as the MIT CRISPR design tool, is something that we sought to test. A score of 100 in the literature implies that there is no predicted on-target activity for this particular guide, and a score of anything less than 70 implies that there are many predicted off-target sites, or at least there are several severe off-target sites. The way this score is calculated is based on the sequence of the guide itself. Some sequences are more prone to have off-target effects than others, and in other cases, generally the scoring function you want to be optimizing for 100. Interestingly, we saw that low specificity caused increased depletion, and so for a knockout study, you would definitely want more depletion. The real salient question, though, is where is that depletion happening and why is that depletion happening? And so low specificity does cause increased depletion, but it's not necessarily something that you want to take forward into your knockout library. You don't want to take forward those guides into your library, particularly because they may be targeting more than one essential gene. Effectively, a low specificity causes increased depletion simply because you were designing a very promiscuous guide, which could be cutting at many points throughout the genome. And so as a predictor, it's difficult to use this Hsu score as a positive predictive measure of depletion in a knockout library design, but best practices imply that we should generally be keeping that score as high as possible, and so we should be avoiding creating guides that target essential genes that have that low off-target score. So moving on to the next slide, 22, just a further note on specificity. We have modified the Hsu score for our library design purposes. This is not something that you'll find in DeskGen Cloud. This is something that is exclusive to our library design, where we've modified the original version of the algorithm, which is a heuristic algorithm, in that it doesn't find every single off-target hit, but rather makes assumptions about the number of off-target hits. and we've turned that into a brute force algorithm which can find every single off-target hit. The reason this is not available on DeshGen Cloud is simply because it is computationally very expensive to run that sort of analysis, but it's something that we can spin up an extra server for when we are designing a library. And so that's part of the standard practice of designing a library and optimizing for specificity, essentially so that we can determine all possible weaknesses in our design by looking for all possible off-target hits and designing that library around them. So moving on to the next slide on sequence properties. So here we looked at two things. First, we looked at the GC content. And what we saw, our findings were concurrent with the Wang et al. 2014 study, which itself suggested that you want to optimize a guide to have about 30 to 70% GC content. Anything outside of that would reduce guide activity. And indeed, you can see from our data here that we saw roughly the same thing. In our case, the data suggested a 40 to 75% appears to be optimal. And so again, this is something that we factor into every guide that we design. And the other thing that we looked at on this slide was homopolymers. So here what we saw is that a guide which had more than 4-length homopolymers appeared to, so anything more than 3 appeared to have much less activity or was had less depletion activity. And so in every case where we're designing one of these guides, we are monitoring to make sure that these homopolymers are less than 4. So on to slide 24, host genotype. This is an interesting thing which is quite surprising how many people who have just got started in gene editing may overlook. So as everyone knows, every model genome, every reference genome is completely different from the model that you're actually going to be working with in the lab. Whether it's a cell that you're working with or a plant or an animal model, all of these genomes are going to vary from the reference genome. And so when you're designing guides using a reference genome, you may be overlooking certain SNPs and other small changes that can definitely impact sgRNA activity. So initially we looked at this. We pulled the data for the A375 melanoma cell line genome from the COSMIC database. And we demonstrated that there were a large number of SNPs and copy number variations. And then in parallel to the study that I've mentioned on slide 25, we decided to introduce a 1 base pair mutation randomly into the protospace of 100 guides that we had designed. The goal of which was essentially to replicate or model this kind of difference between the guide as it was designed and the genome as it was implied from the reference genome. And what we saw is that 98% of sgRNAs that have a single mismatch in the seed region almost completely lose their activity. And so this can be quite serious when you're designing a library or just a general CRISPR experiment. If you fail to factor these issues in and you are designing into a region with many of these so-called SNP collisions, then you could be designing for targets that simply aren't there, or you're designing a guide that is going to have a very different activity profile because of the various SNPs throughout the entire genome. So there's an impact on both the on-target effect and also the off-target effect, which can be quite easy to overlook. But that is something that we do factor into our library design. And we do that by recommending a full whole genome sequencing workup of the model that you're working with prior to the design of the library itself. So moving on to slide 26, I want to briefly touch upon the effect of percent peptide. So what you're viewing here is a small readout from our guide picker tool. Again, you can access it at guidepicker at deshgen.com/guidepicker. The graph on the right shows along the x-axis the percent peptide, and this represents the position of a guide within the total percentage of a peptide. So if you took all of the transcripts and then you pulled the coding DNA sequences from each of those transcripts, concatenated them together into a master coding DNA sequence, and then you chop that into fragments, that would be your percent peptide score. And what you see when you model the on-target DENCH 2016 score against percent peptide is a drop-off in DENCH on-target activity right towards the 5' end of the gene. Sorry, towards the 3' end of the gene, which is quite interesting. And as there are two different versions of the DENS 2016 scoring function. There's the DENS 2016 full score, which takes into account this drop-off towards percent peptide based on the percent peptide, and there is the positionless version of the DENS 2016 score, which does not. The reason I bring this up comes to light on the next slide, slide 27, where we see that in our study there was really only a negligible effect of the percent peptide location on sgRNA efficiency. And this is this really counters some of the dogma of the CRISPR community, where for many years people have been saying that you will get a better depletion, a better knockout by targeting guides towards the first third of the 5' end of the gene. And we demonstrated in that study that this does not appear to be the case, though if you look towards the 3' end on slide 27, you can see that you just begin to see that drop-off, which is reminiscent of the effect that you saw on slide 26. So it does appear that targeting guides right at the 3' end is something that should be avoided, but targeting guides towards the roughly 85% 5' end of the gene is something that can work. And so for us at Desktop Genetics, this is really, really quite exciting because it opens up the guide RNA design space to be tremendously rich. Because we're able to dispense with this dogma, it means that we can design a guide almost anywhere in the gene and still expect there to be activity and depletion. But then this also raises a sort of counterpoint, which is, well, if we can design guides at any point in the gene and they can still be effective, then how do we narrow them down, right? If we find that every guide has the same on-target score or the same off-target score, are there other steps that we could take in order to increase the efficiency of our screen? And the answer, of course, is yes, and that is on the next slide where we discuss target environment, specifically functional domains. So Xie et al. In 2015 demonstrated that by targeting functional domains, they were able to enhance the depletion of a gene and by a specific guide. And this is something that we can definitely employ for our clients, but as with every other scoring function or every other sgRNA design parameter, we did want to test it, largely because functional domains can be quite varied. They can be difficult to deal with. The predictions for them aren't always 100% accurate. And if you look at this data from the PFAM database, what you see is that the functional domains in orange can be sparse. They can be quite sort of sparsely spread out throughout the gene, or they can be almost entirely conserved throughout the entire gene. And so in some cases, you can have guides that have good overlap with a functional domain for a particular gene, like the case of RPL8 on the left, or you can have guides that just overlap with almost every functional domain, like with CCDC84 on the right. So in any case, once we had got started on our study, we looked at the PFAM database, and we did test to see if targeting functional domains produces guides with increased depletion levels. And on slide 29, you can see this, where we showed that by targeting a functional domain, we were able to get a better fold change. And so this is something that we now do factor into our library design. Though again, I do stress that this is something that we must do on a case-by-case basis. And typically our bioinformaticians will first have a sit-down with you or your team to discuss whether or not this would be appropriate in your case. So now on to slide 30, just to touch upon a few other scoring functions. I've mentioned DENS 2014, I've mentioned CHARI 2015, which were comparable. And I've also outlined how DENS 2016 was a significant improvement above those two scoring functions when it came to designing a library for knockout. And we also looked at the Bay 2015 microhomology score, and we saw that it was not something which was a positive predictor for designing guides for a knockout library. But again, I stress that this was not what that scoring function was designed for. If you do want to play with that scoring function yourself, you can take a look on GuidePicker. And then also the Lorenz 2011 for the Lorenz-Vienna RNA fold calculator. We saw that once again this was not a scoring function, which was a positive predictor of knockout activity for a particular guide. So now moving on to slides 31. I want to tie this a little bit back to machine learning, and I want to talk about negative predictors and positive predictors for training a machine learning data set. And again, that is something that we do focus on tremendously at Desktop Genetics. That is what our tech R&D team was designed to do, as well as to design libraries. What we see is that generally at Desktop Genetics and for the field as a whole, currently we are better at determining the guides that will not be useful for a knockout library or for a particular use case. And so what we've seen is that NAG PAMs, GC content, homopolymer numbers, SNP collisions, as well as other things which I haven't gone into detail in on this study, like the uracil triplets and non-constitutive transcripts. What we see here is that these parameters are useful for being negative predictors of using these guides. So when we evaluate these scores, we can essentially, when we evaluate guide candidates using these scores, we can be very confident that they will not be useful in the assay, in the screen or the library that we're designing. On the other hand, if you look at the positive predictors, you can see that we've got more of a mixed bag on the next slide. So essentially here, what you're seeing is that there's only a few scoring functions, CHARI 2015, DENCH 2014, and DENCH 2016, with which we can say quite definitively are useful for evaluating whether the guide will be useful in the screen itself. And all of these other factors either are not useful, for instance, targeting a guide to the first third of the CDS, or we've seen some mixed data on, for instance, the functional domain targeting or the Su et al. 2013 score, which in some cases, we've seen as an essential thing to include in the guide design process, but in of itself is not particularly useful for designing a guide for a knockout. So it's just something that is interesting to bear in mind. It's one of the challenges that we do face at Desktop Genetics and as a CRISPR research field as a whole. But then just to wrap it up on slide 33, I've got 2 left. What we have seen is that when we tie together all of these different sgRNA design parameters or sgRNA scoring functions, when we put them all together into a single stack where we're taking the best negative predictors, the best positive predictors, and we really focus on those, what we see is that we are capable of designing a library where there is a significantly increased fraction of guides that can exhibit more than 2-fold depletion versus competitor libraries. And those competitor libraries, by and large, are the off-the-shelf libraries that have been designed using a reference genome or a certain set of rules. When we go back and we look at the fraction of guides exhibiting more than 2-fold depletion in the initial manual tests that we had seen, only 25% of these guides would exhibit more than 2-fold depletion, which alone was better than the Gecko V2 experiments, and was then later surpassed by the Wang library. But what we've seen at Desktop Genetics is that these libraries have a significantly increased fraction of guides that exhibit more than 2-fold depletion. And as we continue to generate more data in our internal studies, and as we continue to learn more from the field as more and more CRISPR publications come out, we only expect this number to increase and to get more specific as we look at the different experimental intents and use cases of the CRISPR technology itself. For my final slide, just to wrap up on library design, we are guided by the literature to help guide you from design to data. And what we look for is DENCH 2016 score, in some cases CHARI 2015, and DENCH 2014. We're looking at the Su 2013. We're looking for functional domains. We're looking to optimize around GC content, and ideally to conduct these SNP collision analyses by conducting whole genome sequencing prior to library design. And we're also looking to optimize around homopolymers and uracil triplets. What I can say is that the benefits to someone who'd be considering one of these libraries is that you can effectively hand over the guide design problem to experts who've been doing this for years, and you'll have an additional level of independence and unbiased guide design for your experiment. We can genotype the model to get a more effective library. And everything that you're seeing here is driven by our machine learning work, with the goal of ultimately giving you a very rapid turnaround on a novel library design, which will be designed by us and synthesized by Twist. And again, it is a very, very flexible pipeline. You don't have to take advantage of all of these together. You can have just a few. It can be a very small targeted library or genome-wide. Really, it's all down to everything that you need. So with that, I would like to thank you all for listening. Once again, to thank Twist for inviting me. It's always a pleasure. And for Front Line Genomics for hosting. Thank you.
So, thank you very much to both Charles and Edward for your presentations today. I also apologise for a slight lag that we had in slide transitions there, and thank you all for your patience on that one. What I would love to do is invite some questions from the audience. If you have any for our speakers, you can see that there is a box, a Q&A box that you can type into. So if you pop your questions in there, and we will be answering them. So a question that came in during the presentations that I would like to put to you, Charles, to get things rolling, was from Shuo Wang at the MD Anderson Cancer Center, who asks about the length of your oligos. Perhaps that's a question that you can address to begin with.
Sure. Our standard offering allows for oligos up to 150 nucleotides, but if you have anything in the range from 151 to 200, while it's not our standard offering, please feel free to reach out to us, and it's something that we could discuss and evaluate.
Excellent. And so on a similar sort of question again, one for you, Charles, again on the oligos. I mean, how do your oligo pools compare with, say, something from a custom array? Compare with custom array.
Sure. So from some of our early collaborators, what we've seen is that our error rate is significantly lower than what they found in a custom array library, and there's much better representation in our pool versus a custom array pool based on data and feedback that we've been given from our early collaborators.
Fantastic. And so, Edward, turning to you, if I may, I was hoping you could tell us a bit more about the difference between DeskGen and your libraries. I know it's something you touched on during your presentation, but perhaps you could tell us a little bit more about that.
Yeah, no problem. This comes up quite a lot. So you can think of the DeskGen platform as everything that you need to get started with CRISPR, right? It's combines some of the most elementary scoring functions. So in the knockout mode and knock-in mode, you have access to Dench 2016 and the SU 2013 scoring functions, and as well as GC content. And you have the graphical user interface to quickly design a few guides that you might want to sort of use to get started with CRISPR. Typically, I would recommend this to the CRISPR pioneers in the lab, as I refer to them. Effectively, these are the people who have been tasked with setting up the CRISPR workflow for a specific lab. They don't want to get into this horrible mess which we've seen in many of our users, where they have every single tool open all at the same time. Lots of different URLs, lots of different tools spinning up and scoring guides at any particular moment. And then it creates quite a logistical nightmare to tie all of that data together. So DeskGen solves that problem, right? It just lets you get started quickly without having to mess around doing a lot of URL bashing. Guide Picker is also something which is useful. Here you've got 10 scoring functions altogether. So I mentioned microhomology. We've got the 3 different versions of the Dench scoring function, the SU score, GC content, and a few others. And again, here you are using these tools primarily to save time, which otherwise you would waste using some of these slower tools from academia. The library design is very, very different, and you can think of this in the sense that it would be really difficult to design a library using the DeskGen platform or most other CRISPR tools out there. And so it's not impossible. It is something that you can do. But in the case where you don't have a lot of CRISPR expertise or you have just got started with the technology, but you want to move quicker than what a pioneer might set up for you, then I would recommend the library service for these groups. And again, it's not necessarily something which has to be a genome-wide library. You don't have to be ordering a few thousand guides from us. It can be much smaller. It can be a very small, focused panel of a few dozen guides. And so really, it's quite flexible for your needs. So to summarize, DeskGen Cloud, that's what you need to get started. Libraries, if you don't have time to learn the technology and you just want to dive into an experiment or a screen, and the library service is for you.
Fantastic. So we've had quite a few questions about the DeskGen platform come in. So I'll jump in here with one from Stephen Allen, who asks: is there any evidence that the drop-off in efficiency in the 3 UTR is caused by the SNPs that you spoke of, i.e., between reference and experimental genome?
You know, I'm not sure what the answer to that is. I've discussed it with a few people in the past, and there tends to be a mix of responses on this. One of my colleagues was just out at Cold Spring Harbor, and he said that when he presented this slide, this was the one that got the most attention, because at the moment it is still up for debate, so to speak.
Fair enough, fair enough. So coming back to you, Charles, if I may, and again, wondering how the representation of oligos, how it impacts upon screening efficiency.
Sure. Well, if an oligo isn't represented in your pool, it's really missing out on data and a potential hit that might be of value to you. So when you have a very high representation of your oligos, that's also very even, you're going to be able to sequence less. You're going to be able to get all the data that is in the design of your library. And that way you don't have to go back and potentially do the assay again with the same designs and hope that you'll get some data from the designs that you were interested in originally.
And so we've had another question about your oligosynthesis platform in from Yuan Jiang, who asks whether you're able to incorporate you at certain positions of the DNA oligos. So
our platform currently cannot support the incorporation of uracil. It's just your standard DNA bases, but that's something in the long-term future that is possible with our platform. It's just something that we haven't focused on today.
Okay, and so how can your oligo pools help to ensure specificity?
So I think this is a solution that's kind of a joint solution between Desktop Genetics and Twist, where they've got their algorithms that really take into account not just the published material, but also their additional brute force method that Eddie touched on to make sure that there aren't any non-specific targets and make sure that there's the maximum on-target activity, as well as our synthesis. If the more errors there are, the more potential for drop-offs and lower activity in guides that you'll have. So being able to have a really optimal design, as well as maintaining the accurate sequence during synthesis, really benefits the specificity.
Yeah, I'd agree with that. I'd say that the oligo and the quality of the oligo is really the vehicle for the design itself, and it's ultimately the design which is what is responsible for ensuring specificity, but it's also the work that you put in prior to that design. And so if you are working in a primary cell or a cancer line or another cell line that is more mutable than others, then we recommend doing that whole genome sequencing workup, even if the data is available from a database, because we know that there can be other SNPs, there can be other changes in the genotype during the general experimental process. And so if you are prepared to do that work prior to the design, where we can definitely guarantee using the state-of-the-art to ensure that it is of a very high quality, then really Twist is are the guys who then carry that forward, and just by nature of their platform being so accurate, means that those designs and that work that was conducted prior to synthesis is carried forward into the delivery of a library.
Fantastic. So we have a question from Nicholas Sandoval, which I believe is directed more at you, Edward, but Charles, please do jump in if you have something to add, which is, are there robust capabilities for CPF1 designs, such as the one that you described for Cas9?
Are there robust capabilities for what, sorry?
CPF1 designs, such as the ones that you described for Cas9.
Oh, juicy question. Thanks for that. So CPF1 is very interesting. One of the issues that you see with all of the CRISPR nucleases, whether it's SP-Cas9 or NM-Cas9 or CPF1, is that they're relatively new. Right? These things have only come out in the last few years. The most well-characterized nuclease itself is SP-Cas9. That tends to be the workhorse of most people using CRISPR. And one of the reasons for that is because there is quite a, how to put it, there's a good community creating scoring functions for that kind of Cas9. And so just recalling Dench and Sue and Chari, these algorithmic models were trained on data that was generated for that Cas9. Right, so Dench looked at, I want to say, he looked at it in 2014. It was a few thousand guides designed for human and mouse to characterize the activity of SP-Cas9. But if you take that scoring function and then you apply it to the other Cas9s, there's absolutely no guarantee that it will be as predictive. Further, if you take a scoring function that was designed to optimize SP-Cas9 in humans and mice, there's no way that you can guarantee that it would optimize its performance in, say, ZMAs, completely different organisms. So when you look at CPF1, which is a different nuclease, and really has only come to light in the last, got a lot of serious attention in the last year and a half, it's still quite young, and so there are robust capabilities for it in that, the PAM sites are different, so as a result you can target different sequences, but the nascent scoring functions that have been developed for it haven't really materialized in it as meaningful way. So we're still waiting to see the characterization for all of the performance data on CPF1. Some of this is starting to come out, and this was recently discussed at the Cold Spring Harbor Lab meeting as well. So I'm really excited by where CPF1 is going, and as more scoring functions come out for it, we'll definitely be rolling those both into the DeskGen platform so people can use it more effectively, as well as the library designs as well. Oh, and just one final thing that I'd recommend investigating the CRISPR as well, CRISPR, which has a good amount of data and discussion on which scoring functions are valid for which nucleases in which model organisms.
Thank you. So I'm keenly aware that we're already running over time. So I think we'll make this the final question, which comes from Thomas Quinn. Again, I believe one that's directed towards you, Edward. But again, Charles, please do add anything that you have that you want to say on this question, which is, what is your experience with drop of representation from plasmid library to representation within virus particles?
Good question. Don't have the answer.
Fair enough.
Yeah, and I'm sorry, don't have an answer either. The projects that we're working on with our collaborators, they've gone through their plasmid cloning portion, but are still now working on the actual screen, so we don't have any data for that just yet.
Yep. So I'd say if you're still with us, Thomas, do drop me an email, and I'll connect you with Dr. Lee Brody, who is our resident expert on all things virus.
All right. Well, So that is it for the questions that we have had in. So once again, I want to say a huge thank you to our speakers today, Charles Joseph, and to Edward Perello, and to Twist Bioscience for partnering with us on this webinar. If you are watching this broadcast on demand later on and you have a question, please send it over to myself at Front Line Genomics. So you can get me at liz@frontlinegenomics.com. And just send over a question and I'll be happy to pass it along. Also, all that remains is for me to say, once again, thank you very much to our speakers. Thank you to all of you for joining us today, and enjoy the rest of your days. All right.